This
Start.
Good
conference
morning,
will
sir.
now be recorded.
Yeah, morning. We'll start. Okay, so last session we were covering ICH GCP E6. I hope everyone has downloaded that document. Uh,
Ball देखो, ball देखो. Get me the ball. दे.
Narmada, you're there.
Ball.
Narmada, you're there. Well, right. So now you can see here. We'll start. Clinical trial study. We have started, correct? So clinical study report also we covered, correct? Clinical study report is a document which we submitted to regulatory authority, which contains all statistical descriptions, presentations, analysis. Complete report is CSR. Okay, right. So structure and content of clinical study report you can understand or you can get to know from ICH E3 guidance document. Comparator is nothing but a product which is used for comparison. Okay, so it can be a active control or it can be a placebo which is used as a reference. So comparator means if the product is already available in the market, we do product which is already available in the market. Otherwise, we will use placebo. Placebo means inactive compound. Right. Compliance is adhering to all the trial related requirements. So in compliance with means you are following all the standards. Okay, GCP requirements and other regulatory requirements. Confidentiality is nothing but secret, maintaining it secret, not disclosing things to anyone. Contract means agreement between two parties. CRO stands for Contract Research Organization.
Organization.
Contract Research Organization. Documentation. So all your documentation in a clinical trial data, you will collect all the data in written format, electronic format, magnetic and optical records, X-rays, scans, and ECG reports, electrocardiogram reports. This is all documentation. And GCP, this is important. I want everyone to read this GCP. GCP stands for Good Clinical
Good clinical practice.
Clinical practice.
Good Clinical Practice.
Practice.
Right. So standard for the design, conduct, and performance, monitoring, auditing, recording, analysis, and reporting of clinical trial data that provides assurance that the data and reported results are credible and accurate, and all that rights, integrity, and confidentiality of trial subjects is protected. Okay, so this is a very good way of designing a trial, conducting a trial, performing a trial-related activities, monitoring, auditing. So your GCP is standard way of all the activities that are part of your clinical trial. Whenever you are following standards, the quality of the outcome will be very good. Right? Adhe kada brand. So whenever I say brand means they maintain some certain standards. Correct? Fine. Now you can see IDMC. IDMC stands for Independent Data Monitoring Committee. So this committee has the right to investigate your trial, check your safety data, critical efficacy endpoints, and to recommend. These committee will recommend the sponsor whether to continue the trial, stop or modify a trial. Suppose to say, I have invented XYZ 500 mg. We are giving dose to all the subjects. So in middle of the trial, IDMC will have a verification how the drug is performing, whether it is okay, all the safety data is okay, all the efficacy critical endpoints are matching, yes or no. They will come and cross check. So after they have cross checked, suppose to say out of 30 people, 23 people got adverse events. This is an issue. Immediately they will recommend sponsor to stop conducting the trial. Okay, IDMC is Independent Data Monitoring Committee. So sponsor has no link with this. Okay, sponsor cannot influence this team. So this team will tell sponsor whether to continue the trial, modify or stop the trial. Weakness and తెలుసుగా. IEC stands for
Independent ethics.
Independent Ethics Committee. So we have one more team called IRB. IEC, IRB, both are one and the same. Okay. So Independent Ethics Committee. So whenever you are conducting a clinical trial, whenever you are conducting a clinical trial, you have to take approval from this IEC. IEC stands for Independent Ethics Committee. Before you are conducting a clinical trial, you have to take approval from IEC. IEC is Independent Ethics Committee. Okay. Because I worked in initially I worked in a very small organization, so I know the all the process. When you are working for an MNC, straight away they will ask to open SAS, do that data set, do it. So in a small organization, you will have a very good scope to have a complete exposure about your drug development. So whatever the company I work there, we have formulations development, FR&D is there, AR&D is there, bioanalytic clinical development division is there in the same office. In my office, they used to recruit patients, so they used to give drug to the patients. They used to collect data. Clinical development division, they do. And whatever the drug samples they have collected, we have bioanalytical team. So bioanalytical department, in that bioanalytical department, they will analyze your drug bioavailability, how much concentration is present in your blood samples. They will do bioavailability. They have PK team, PKPD team. So PK team is they build a pharmacokinetics team. So there we have statisticians, statistical programmers, and what all the results they do, they will give it to finally end sponsor. Medical writing team will prepare documents, CSR, they will submit it to the team. Got the project, got the concept. So this is a drug development cycle. These many departments are involved, when we are developing a new drug. So I worked in a small organization. So there we have all these facilities, all these features we have. So the reason, I'm pretty much aware of the process. Not everyone who is working in real-time scenarios will know this much of details regarding the process. Regarding the process, they are not sure. Okay, right. So IEC is, Independent Ethics Committee. So whatever the protocol we do, that protocol synopsis we will submit to IEC. So your IEC is a group of members. Okay, IEC is a group of members. It is a group of members which contains medical and non-medical members. Okay, in IEC you will have a layman, worker, religious priest, you have lawyer. Okay, so you have a data manager, you have a group of people. Okay, so all these people together, they will come to an understanding, you know, we can use this drug, yes or no. A lawyer also will be there because, if supposed to say if you are using cocaine in your medicine, huh? Is it it is allowed in India, cocaine? No, right? So obviously, so the reason lawyer will be there, priest will be there, so layman will be there, social workers will be there. So this IEC is a group of multiple people. Huh? పూజారులు వాళ్ళు కూడా ఉంటారు. సెంటిమెంట్స్ ఉంటాయి కదా కొంతమందికి కొన్ని థింగ్స్ మీద, అలాంటివి కూడా చెక్ చేస్తారు. ఓకే, రైట్, ఫైన్. So you see, right, everybody will be there. Okay, right. So all these people, they will review our synopsis. Okay, they will review our synopsis, and if they find everything is good, then they will give approval. After their approval only you are allowed to start clinical trial. If they have said that, like suppose to say in my protocol I have written XYZ 1000 mg. So who will do step here? I will do straight away. I will dose 1000 mg to patients. So if you are doing 1000 mg to patients, what happens? It will cause severe side effects, right? Because human body cannot adjust more than 500, 600, correct? Nowadays we are adjusted to 650, correct? So ఇండియాకి అందుకు 2000 3000 కూడా పోతుంది, right? So that is how it is, you see, right? um, So in this case, IEC will give a point, audit point saying that, boss, I will not allow to dose 1000 mg because this drug can cause side effects, adverse events. Okay, so do you have any strong proof that it works with 1000 mg? I mean to say any comparator product which has already tested with 1000 mg. So if you have, bring me that data. Otherwise, I will not allow you to conduct on 1000 mg. So I will insist you to go with 250, then increase to 500, then increase to 650, but not 1000. So likewise, they will give audit points. Based on them audit points, they have to correct it and they have to finalize the protocol. Once protocol is finalized, after their approval from IEC or IRB, you can start conducting a clinical trial. Is that fine? Right. So informed consent is whenever you are going to a hospital, nowadays everybody is taking informed consent. Right? Informed consent is in prior they will tell all the activities. Okay, suppose to say recently I have taken my kid to a dentist. Okay, there they have given informed consent. So suppose to say if suppose to say if you have a tooth decay, they will do root canal, they will do caps, right? They'll fix a cap, right? So in the informed consent it is clearly written, cap may come out, or you may see that chipping may happen to that cap. So అన్ని డబ్బులు తీసుకుంటారు మళ్ళీ అన్నీ అవుతాయంటే who will do here? So it is they says that is our process. If you want you go ahead, otherwise tomorrow they don't want to anyone to you know comment that your treatment is not good, or nobody can go legally, correct? Because they have officially signed the document, right? So when anybody is going for a surgery, they will take approval, they will take informed consent, correct? Why? సర్జరీలో అటు ఇటు అయితే మాకు సంబంధం లేదు, right? So if something happens in surgery, we are not responsible. That is what we try to do good. If it is good, absolutely fine. End of the day, everyone are happy. If something is wrong, you cannot claim that we killed that guy. So that is informed consent. If you are okay for that, then only we will go. Otherwise, no. Correct? So informed consent is a document which contains complete information about a clinical trial. So what happens to the subject if you're taking, so how much amount they will give, everything, like they will write. If suppose to say if you're taking this XYZ 500 mg, you may feel nausea, you may feel skin rashes, maybe like that. So they will clearly tell all the issues they have seen on animals, and animals they have seen issues, right? Correct? So they will write clearly saying that maybe if you're taking this medication you may face so and so. Okay, right? So sponsor, like your subject, after reading all these things, if he is still willing to participate, he has to participate, right? Next, inspection is nothing but checking the things. IRB, I told you, right? IEC, IRB, both are one and the same. Okay, right? And interim analysis. Interim analysis is important. I want everyone to focus. If you want, you can note it down, because there is a chance people might ask you if you have worked on IA, interim analysis. So what is interim analysis? Suppose to say if I am conducting a clinical trial based on 1000 subjects. If I'm conducting a clinical trial based on 1000 subjects. Okay, all your clinical trials are conducted in double blinded manner. All your clinical trials are conducted in double-blinded manner. What is a double-blinded?
Both they do not
The
know.
subject and the
Patient and
investigator,
investigator do
they
not
Both do
don't
know.
know
not
what
aware
is
about that.
administered,
Right.
what drug is
Both,
administered.
correct. So both the subject and investigator are not aware what
Not
treatment
aware
they
of.
have given. Correct? Right. So now in interim analysis, whenever we receive 80% of clean data, because when whatever the data they capture, you will find lot of data issues in clinical trials. Suppose to say start date is greater than end date. Can start date be greater than end date? No, right? Suppose to say there are cases where you see start date greater than end date. Suppose to say adverse event term is missing. This is a required variable. It cannot have missing. Okay. Suppose to say I say subject number one got adverse event. But still, what is the event? The subject has got that information should be there, right? If that information is not there, how should I understand whether the subject got adverse event, yes or no? How will I understand? So if you have any missing values in required fields, so all that these are all data issues. So you may get a lot of data issues when working on clinical trials. Okay, right. So now uh, once we have received 80% of clean data. So 80% of clean data means this 800 subjects, whatever data we have right now, there are no data issues. This is a final copy of your data. So now in this case, అర్హత ఉంటుంది కదా, your sponsor, your investigator will have always like they are very much enthusiastic, right? Whether my drug is pass yes or no. They wanted to check, correct? Particularly in phase 3 trials, they will have a lot of enthusiasm. So in this case, your regulatory authority will give you a opportunity. The opportunity is uh, so till this 80% data is collected, all your randomization codes are blinded. All your randomization codes are blinded. Blinded means, so whether the subject has taken, whether the subject has taken treatment A or treatment B, nobody does not know. That is what blinding, correct? So till then they are blinded. So once we have received 80% of clinical trial data, you can take original randomization, real randomization. You can take real randomization and you can do the analysis. But this analysis you cannot perform, but this analysis you cannot perform, but this analysis you cannot perform by the person who is already participated in the clinical trial. So an independent programmer and independent statistician will come into your study. An independent programmer, independent statistician. Whenever I say independent programmer means this programmer has never worked in your study, is completely from outside. So when this independent programmer and independent statistician will come into the study, they will run all your safety and efficacy analysis with original randomization. Original randomization. So whenever they are running the data with original randomization, they can see whether the drug is working fine, yes or no, right? Your biostatistician is the one who can understand the final conclusion, yes or no, yes or no. So they can do it. So why your regulatory will allow you to do this? Why your regulatory will allow to do this? So this concept, why your regulatory will allow to do this is, so suppose to say whenever you are working on a clinical trial, so your clinical trial sometimes you may get result in 1 year, 2 years, 5 years, even it may take 10 years also. So what all the analysis you have done during these 10 years, I have to submit it to regulatory authority. So first I conducted trial one. So I failed. So I modified my drug like this. Now I have done this. I'm failed. Again I have modified my drug this way. I failed. So 4 or 5 instances right from every beginning you have to give the document. You have to give the complete document to regulatory authority. So to get a 10 years data, how much time it will take here? At least 3 4 months. 3 4 months అయితే పట్టుద్దా లేదా? So before 10 years means I'm not there here. I'm not there. I just joined this organization 1 year back. So how can I give 10 years data here? So everything I have to ensure, right? Now I am there, people will ask me, correct? So I have to check where that data is there. I have to organize all the trials, 5 or 6 trials. I have to do it, correct? Correct? Yes or no? So I have to do all these. So for me, it will take some 3 to 4 months time. At the minimum, it will take 3 to 4 months time. Not only for me, for all the departments, it will take time. Correct? Correct? Right. So after doing this, if the trial is success, immediately they will alert all the team, other departments. Boss, our trial is a success. Please make sure your records are kept ready. So by the time I get that 20% data, so they have to prepare that they have to prepare the complete documentation part. So that 20% data takes at least 1 to 2 months, right? So in next 1 to 2 months, we get the data. So we generate results. In 1 or 2 months, we will send all the reports for filing. Okay, at least 2 months before you can get this filing, right? So at least 2 months before we can get drug into the market, right? That is the reason your regulatory will allow to perform interim analysis. Got the point, yes or no? So whenever you are proven on 80%, obviously rest of 20% you are okay. Correct? Right. Okay, so this is interim analysis. Okay, interim analysis కోసం ఇంత చెప్పాను మీకు. Investigational product is the drug which we are testing. Investigator is a person who is responsible for conduct of clinical trial in a particular site. Investigator, institution. Both are same. IB, investigator's brochure. So in this brochure, you have complete information about your drug. Complete information means both clinical and non-clinical data related to your drug will be presented on investigator's brochure, IB. Legally acceptable representative, whenever the subject is a 3 years or whenever the subject is a minor, on behalf of that one representative should sign the ICF. Okay, that is legally acceptable representative. Monitoring means observing whether the activities are going in a right way, yes or no. Monitoring report, generating a report. Multi-center trial, whenever we are conducting a clinical trial at multiple places, suppose to say I want 10,000 cancer patients. Can I get all 10,000 cancer patients in at one site? No, right? So I have to conduct this trial at multiple places, multi-center trial. Protocol, read the definition. Protocol. You are going through the right protocol. Hmm?
Yes, sir.
So describes the study design, methodology, and statistical considerations, and organizing a clinical trial. Protocol amendment. Any changes to your protocol is considered as protocol amendment. QA/QC, quality assurance, is the one who is going to, approve quality on final product. QC is inline, correct? In each and every department, internally you have QC. Randomization is a process of assigning subjects to treatments or control groups using an element of chance. Regulatory authorities, we know. SAE, serious adverse event. You can note down, this is important. Any untoward medical occurrence that has happened may be due to our dose, may or may not be due to your dose, and that untoward occurrence is resulting in death. That side effect, finally the subject died due to that side effect. So should be considered as a serious adverse event. That untoward occurrence is leading to life threatening. Then we need to consider as serious adverse event. Requires inpatient hospitalization or prolongation of existing hospitalization. Is supposed to say, if I have to take the subject to hospital, then I need to consider as a SAE. So results in persistent or significant disability or incapacity. So if the person is not able to, move here and there, so he lost some senses maybe. So that also we need to consider as serious adverse events. Is a congenital and anomaly or birth defect. So if any birth defects has happened due to this, we should consider as a serious adverse events. Right. Source documents means you can see, right, laboratory notes, hospital records, clinical and office charts, subject diaries, evaluation checklists, pharmacy dispensing records, recorded data from automated instruments, copies or transcriptions certified after verification as being accurate copies, so microfeatures, photographic negatives, these are all your source documents. మనం ఇన్ని వాడాలి. So sponsor is nothing but our God. Sponsor is nothing but our God. Correct? అంతేగా పైసలు ఇచ్చేటాడు దేవుడు, right? So an individual, company, organization which takes the responsibility for the initiation, management and or financing of a clinical trial. Right. Sponsor investigator is sponsor side, you will have one lead, right? So he is a sponsor investigator. SOP stands for standard operating system. Sub investigator అవసరం లేదు. So a person who participate in a clinical trial, we call it as a subject or a trial subject. Subject ID, subject code, trial site, unexpected drug reactions. This is okay. Vulnerable subjects. This is not it. So you see, right? This is all about your clinical domain terminology. Okay, clinical domain terminology. Right. Next. So once we have conducted clinical trial, we have to prepare CSR. That CSR you need to submit to regulatory authority, right? So in which section, what information we have to present is given in, ICH E3 guidance document. You see, right? Title page, you should have a title page, you should have synopsis, you have table of contents, list of abbreviations used in this, and ethics, IEC, IRB approval. Because first of all, you should say, I have taken approval from IRB, IEC, then I conducted a trial, then these are my study objectives, this is my investigational plan, and these are my inclusion exclusion criterias. This is what I wanted to test as per study design. These are all the treatments. These are all your efficacy and safety variables, and whether the safety and efficacy are satisfied, yes or no, I'm applying my statistical methods. And finally, I have to give this information in section 14. You see, right? Section 14 is our important section. So 14.1 is demographic data. So whenever we are generating tables, these tables we generate in section 14.1, 14.2, 14.3, whatever the tables we generate, that will be presented in section 14, 14.1, 14.2, 14.3. Demographic data, efficacy data, safety data. Okay, and related things we keep in 16. Okay, 16 is right. And documentation, whenever you are applying a ANOVA, we get sum of squares, type 3 sum of squares, type 1 sum of squares, calculated value, F ratios, all that backend information will be stored in 16.1.9 documentation of statistical methods. And you will have 16.2 patient data listings. Okay, right. So this is how we present information in a CSR. మనం ఏం చేయాల్సిన అవసరం లేదు ఇందులో. వాట్ ఆల్ ది analysis reports we generate, we submit it to medical writers. Medical writers will take all the information and they will prepare CSR document. Okay, right. బట్ మనం చేసే టేబుల్స్ ఏ సెక్షన్లోకి వెళ్తాయంటే 14.1, 14 section and 16 section, 16.2, 16.1.9. There our information will go in CSR. That is just a basic understanding. And E9, guidance of statistical tests. So I want everyone to go through one point, is right? So section 3.1, 3.1.1.1, 3.1.2. I want you guys to understand parallel design, crossover design. And I wanted you guys to go through 5.2.1, 2.2, 2.3. ఓకే. సో ఇవి చేస్తే సరిపోతుంది మనకి వీటిలో. ఇంతకు మించి అవసరం లేదు. నేను చెప్పినయే 3, 4 పాయింట్లు అవి కూడా మీరు చూడకపోతే నేను ఊరుకొని చెప్తున్నా. దిస్ ఈస్ థియరీ. హూఎవర్ రీడ్స్ విల్ అండర్స్టాండ్. ఓకే. Got the point? Because
Yes,
whenever you
sir.
are working on analysis tables, compulsory you have to select safety population, full analysis set population. సో ఫుల్ అనాలిసిస్ సెట్ అంటే ఏంటో తెలియకుండా మీరు అక్కడ అనాలిసిస్ చేయాలంటే చేయలేరు. సో 3.1.1, 3.1.2, ఓకే. 3.1, 5.1 చూసుకోండి అబ్బా. 5.2, 3.1, 5.2, these two sections I want everyone to go through. Okay, rest statistician headache, missing values and outliers, how to handle them. So your statistician will take care. Okay, right. And 21 CFR part 11, you see, right? This is a 21 CFR part 11. So how to handle with electronic signatures and all, so will be presented here. So clinical trial data, I told you, right? This is a lifetime document. So whatever the document we have got, so all that document you have to convert into PDF. Okay, and that PDF will be digitally signatured. So PDF file, you have to sign the document digitally, signature you have to do it. So that way, that will be present in full version of PDF. You can do that. Is right, signature is there, right? After going through the document, you have to prepare your signature. So you have to add text, Raj. Any text here? So you have to add some text here. So the full version కాదు, full version అయితే వస్తుంది. RAJ Raj, approved by Raj. So you see, right? This is a complete version. So ఇక్కడ approved by Raj అని వస్తుంది. దీనికి ఒక పాస్వర్డ్ ఉంటుంది. So a password you have to give, then only um, you can see, right? You can place a signature there. Okay, right. So ఇంకా ఫుల్ వర్షన్ కాదు. You see, right? Signature వచ్చేసింది చూడండి. Place a signature, approved by Raj. దానిపైన signature, place a signature అంటే you see Raj is with. ఓకే, రైట్. సో ఎక్కడ సిగ్నేచర్ పేజ్ చేయాలి, ఇక్కడ పేజ్ చేయాలి. ఎక్కడ కావాలంటే అక్కడ మనం సిగ్నేచర్ చేసుకోవచ్చు. ఓకే, సో ఇలా ఎలా పడితే అలా చేయకుండా మీకు ఒక డాక్యుమెంట్, ఒక పాస్వర్డ్ ఉంటుంది. సో ఇఫ్ యూ గివ్ దట్ పాస్వర్డ్, దెన్ ఓన్లీ యూ విల్ బి ఏబుల్ టు డూ దిస్, అదర్వైజ్ యూ కెనాట్ డూ ఇట్. ఓకే, రైట్. So that is how it is. So this is about 21 CFR part 11. Okay, right. So I'll stop this here today. Tomorrow I will start with CDISC concept. Tomorrow I will start with CDISC concept. Clear to everyone, yes or no?
Sir.
Right. Okay, that's all for today, guys. Thank you. So this is just a basic introduction about clinical trials. So I have told you the departments also, right? FR&D, AR&D. After that, that information goes to clinical development division. After that, it goes to bioanalytical team. After that, it goes to pharmacokinetics and biostatistics. After that, it will go to CSR. Medical writers will take that CSR. That CSR will be submitted to RA, regulatory affairs activities. So all that together, they have sent the document CSR to your regulatory authorities. Okay, right. Okay, that's all for today, guys. Thank you. Bye.